We are going to discuss a paper that shows senolytics, agents that kill senescent cells, can actually reduce brain damage caused by global cerebral ischemia. Global cerebral ischemia is caused by events such as a cardiac arrest, like a heart attack, situations in which blood flow to the brain is stopped. It's a major problem. About 3 million people a year die from it, and about 3 million people or more become permanently disabled. This is caused by a progression of events, inflammatory cascades, self-feeding inflammatory cascades that cause progressive brain damage even after blood flow has been reestablished. And this is why in many situations of injury we see an increase in senescent cells. The question is, does global cerebral ischemia cause an increase in senescent cells? That's the question of this paper. And the answer is yes, and the answer is if you kill the senescent cells with senolytics, you can improve the outcome of global cerebral ischemia. So to dive into the data, what they did is they induced global cerebral ischemia in the mice, and then they looked at the number of astrocytes. Astrocytes are cells in the brain that too many astrocytes are associated with inflammation. As you can see in the top right figure, there is sham control. That's a small number of astrocytes. Once you induce global cerebral ischemia, the number increases. Once you treat with dasatinib or quercetin, which are senolytics, once you give the senolytics, even like 12 weeks after the insult, you can see a decrease in the number of astrocytes, and in the lower right panel, you can see a decrease in the number of senescent astrocytes as quantified by the senescent marker P16. The same is seen when you look at microglia. Microglia are the inflammatory cells, macrophages of the brain. The global cerebral ischemia induces an increase in these cells, and then when you treat with the senolytics, you have a decrease in the number of infiltrating microglia, indicating reduced inflammation, and in the number of the decrease in the number of senescent microglia as detected by P19. When you look at inflammatory cytokines, so these are the chemical mediators that cause brain damage overall, you can again see the pattern of global cerebral ischemia induces an increase in these inflammatory markers, IL-1 beta, IL-6, and TNF, and the treatment with senolytics decreases it. Most importantly, in the memory test. So this is an interesting test in which sham, you can see the mouse remembers where to go. That's the tracing of how the mouse remembers in a secondary exposure to this maze. The global cerebral ischemia, you see the mouse does not remember, it's looking around. There's a lot more mouse movement because doesn't remember where to find its target. And the pre-treatment with dasatinib or quercetin improves the spatial memory. I encourage you to read the paper. Obviously, I'm just giving you a snapshot here. The paper's in the link below. And so in conclusion, global cerebral ischemia causes generation of senescent cells. Senolytics agents, in this case dasatinib or quercetin, that kill senescent cells, they actually reduce the number of senescent cells and the inflammation associated with senescence. And in this case, administration of senolytics was able to improve memory. Thank you very much.